What are the functions of helper T cells and cytotoxic T cells and cell-mediated immunity? The two functions are the helper T cells and the cytotoxic T cells. The MHC2 is recognized by the CD4, and the helper T cells activate cytokines and macrophages, while the cytotoxic T cells is recognized by CD8+, which recognizes MHC1. And cytotoxic T cells directly target infected cells. What are the steps in the activation of T lymphocytes? The steps of activation is, the first step is antigen recognition. The second step is cytokine secretion and cytokine receptor expression. And the third step is proliferation. The fourth step is differentiation. And the fifth step is effector functions of the helper and cytotoxic. What signals are needed to activate T lymphocytes? The signal that is needed to activate the T lymphocytes is the interleukin-2, which could be seen in step 2 of activation. What cellular receptors and ligands are used to sense and respond to activate T lymphocytes? The T lymphocytes use T cell receptors, which are TCRs, to sense antigens recognized by antigen-presenting cells, APCs, on MHC molecules. The interaction between TCRs and antigen trigger T cells activations. Additionally, co-receptors such as CD4 or CD8 aid in stabilizing the interaction between T cells and APCs. So here the surface molecule of the T lymphocytes is CD3, and the function is the signal transduction by the TCR complex. And this another major one is CD4, which does signaling transduction. And CD8 is signaling transduction's function as well. And the name of the CD4 ligand is class 2 MHC, which is expressed on the antigen presenting cells. And for CD8, the ligand is class 1 MHC, which recognizes all nucleated cells. CD1's function is inhibition, and its ligand is PDL1, PDL2, and it's expressed on the antigen-presenting cells, tissue cells, and tumor cells. While the LFA1's function is adhesion, signal transduction, and the ligand's name is ICAM1, which expressed on the antigen-presenting cells of the endothelium.
What are the types of T cell mediated immune reactions? The two types of T cell mediated immunity is the cytokine release, which is enacted by CD4 lymphocytes, and apoptosis, which is enacted by CD8 lymphocytes. What are the subclasses and their roles in cell-mediated immunity? The subclasses of CD4 lymphocytes are T helper 1, which has a role in macrophage activation by releasing interferon gamma. This is known as the classical pathway of macrophage activation, and it also releases interleukin-2, which aids with the inflammation. T helper 2 lymphocytes, they release interleukin-5, which help with the activation of eosinophils. This is helpful in case of a parasitic infection. And it also releases interleukin-4 and interleukin-13, which takes part in the alternative pathway of macrophage activation. And lastly, the T helper 17, it plays a role with inflammation by releasing interleukin-17 and enforces the epithelium cells by releasing interleukin-22. What are the responses of macrophages that result in the killing of ingested microbes? After being activated, macrophages have three responses. The first one is that they produce reactive oxygen species, nitric oxide, and lysosomal enzymes, which kill the microbes in the phagolysosomes. They also secrete cytokines such as TNF, interleukin 1, and interleukin 12. And they play a role with differentiation of the T helper 1, production of interferon gamma, and TNF and interleukin 1, they play a role with inflammation. And lastly, they express B7 co-stimulators, which play a role with T cell activation. How do CD8 TCLs kill cell infected with viruses? So first, they recognize the virus antigen through the MHC2, sorry, with MHC1, through the CD8 receptor. They also have this co-stimulator, which is ICAM1, react with a receptor LFA1. After this happens, the CTL activates granule exocytosis and basically liberates these two enzymes called granzymes and perforin. Perforin facilitates the entry of granzymes into the cytosol, while granzymes activate apoptosis by cleaving gas bases and activating a pathway that in the end leads to apoptosis of the entire cell.